Good morning, everyone, and I'd really like to thank Gargi College for hosting this event, your principal, Dr. Tyagi, and Dr. Chaturvedi for having us here. I must confess that I've never been so nervous in my life because it's, at one hand, really fascinating to be able to have an interactive session, and I'll try to make it that, with some of the brightest and the best students in the country. But on the other hand, since science is always a learning experience, I'm not sure how valuable I'll be in that interaction, but let's see. One of the challenges we have in India is why, to answer the question, why are we doing science? Yeah. Maybe I should stand a little further away from this cable. Yeah, and historically what's happened in the way science has been done in India is that it started off at a time in the early 20th century when British colonialism was well established, and the British started putting in place presidencies whose purpose was meant to train people, usually men, unfortunately, to govern the British Empire. Some people subverted this purpose and took on interesting questions in science and excelled compared to the best people anywhere in the world. And these were people such as pioneered by Jagdish Bose and followed by several other people in the presidencies of Calcutta, Madras, and Bombay, and so on. This was actually the way science was done everywhere in the world. There wasn't an institutionalized science. Soon after the Second World War, actually just before and during and little after the Second World War, science became much more institutionalized. There were scientific agencies, universities invested in science departments, and science moved from being the practice of essentially gentlemen scientists to becoming an enterprise. And that changed the way science is done all over the world. But even though India established huge enterprises such as the Council for Scientific and Industrial Research, several scientific institutions and research universities, what has happened over the last 60-ish years is that there's an extraordinary foundation of quality science in the country, yet Indian science is known all over the world for the quality people it produces. Indian science is not yet regarded as competitive in world science. Why is that the case? There are many reasons for that. One of it is that the centers of excellence in science are in the West, and it's very difficult for someone to excel, no matter how good you are, unless you're in a highly interactive, competitive environment. And those interactive environments are still far away. But what I'd like to tell you today is, because of this huge foundation which we have in science over the last 60 years, the parts are actually there for a transformation. By connecting these parts together and in some ways looking at problems from within India, Indian science can actually not only transform itself and be a very competitive force in the world, but can actually also transform India. And you are the agents of such a transformation. And if you can manage that transformation through science, or help in the transformation through science, you can not only change yourselves, change India, change Indian science, but because of the huge demography, the huge population of young Indians, you can actually be an agent for transformation in the world. So this talk, therefore, is not about brain drain or reverse brain drain and so on and so forth. It's actually about another topic, which is how we make our brains, and I'll tell you a little story about how that happens, how from an egg you can actually make complex organisms such as mouse or flies or humans, and thereby complex tissues such as the brains, but also that these constructions of organisms from an egg, a complex organism from an egg, is an interaction between genes and environment. And therefore, in many ways, what we are today is shaped not only by our genes, but by our environment. And our environment can be quite a mess. As particularly in India, we have a situation where our nutrition, our sanitation, our health issues, our agricultural issues have actually made India a disaster zone in many ways, in terms of statistics for any disease or any problem. For example, if you look at children under 5 years of age who suffer from stunted growth, India is got a frequency which is higher than that of sub-Saharan Africa, right? So India, strangely, is a mix of a country which has the ability to understand the complex difficulties it has, ability to understand all the science in the world, and an ability to use the science as one of the agents for transformation, right? So we have messed up our world quite a bit. We have messed up our brains and our drains, and we have actually the opportunity to fix both, and people such as you can actually take the lead and are taking the lead. So let's go on now from this to ask two kinds of questions. How did the complex set of organisms such as us come about? So how did nature's engineering construct such complex organisms? And that, as I told you, is an interaction between our genes and our environment, and I'll tell you a story about how that happens. And then, because environment is so important, as scientists of all kinds, how we can actually look at this interaction and both understand nature's engineering, but also understand how the environment impacts on it and how we can change things for the better. So first of all, let's look at nature's engineering. So here are, the dissected model of humans, male and female, and when we try to understand how such complex organisms are made, our brains, our guts, our stomach, our heart, our lungs, and so on and so forth, we as biologists, or as scientists in general looking at biology, ask just three kinds of questions. Where do we come from? How is it that life originated on Earth and resulted in all this complexity? So where do we come from is a question in evolution. The next question is, many biologists ask, is how are we made? And that's a question in embryology or developmental biology. And the third question is, how do we function? And that's a question in physiology. So biologists really, no matter what you call them, biochemists, botanists, bioinformaticians, and so on and so forth, are really of three kinds: evolutionary biologists, developmental biologists, and physiologists. Everything in biology can be put into these three aspects because these are the questions biologists ask. So let's try and answer these three questions quickly. So where do we come from? Now, if you look at life on Earth, one of the mysteries we still don't understand is how the first kinds of replicative forms originated. And that's a big debate and a big puzzle and lots of theories, and I won't go into that. But after that happened, the earliest life forms were on this wall at t is equal to 0 at the start of time of life on Earth, and these were unicellular organisms, let's say, okay? Bacteria, viruses which interact with bacteria and so on and so forth. Simple organisms of various kinds. And the bulk of life on Earth was actually consisted of these simple organisms and still do. Over time, what happened was various kinds of features evolved. And these have, because they have common origins, these features actually are the following. There's a shared chemistry. Since early life started with this origin, a shared origin, all the chemistry of early life forms are seen in many ways in later and later life forms, right? These later forms have this shared cellular chemistry and shared thread of nucleic acid, RNA and DNA connecting this chemistry. And later on, as complexity developed, you had cellular compartmentalization. Different cells had different parts in it which were equally complex. And then multicellularity arose, and then multicellular organisms arose. And these different kinds of so-called complexities, because the original life on Earth, as it were, is actually extraordinarily complex. And these different kinds of complexities in multicellular organisms resulted from a shared chemistry and a shared cellular organization. And that's very important. We know these details actually today at an extraordinary molecular level, but you don't need to look inside to get this answer. And indeed, people such as Charles Darwin and Alfred Russel Wallace looked at complexity on Earth from the outside and came up with the amazing theory that evolution of all this complexity, of all these species on the planet, arose by natural selection, right? And one of the aspects regarding natural selection, which Charles Darwin observed and which is rather dramatic, are these diversities of finches in the Galapagos Islands. So Darwin went on the voyage of the Beagle, and he went to the Galapagos Islands in South America, and there he found that different islands had different species of finches, each with different characteristic beak morphology. And he argued that the isolation of these finches in these different islands resulted in speciation and an exquisite adaptation to their environment and the food they ate, and that's why you had different kinds of beaks. These finches fed on cacti, on other flowers and other islands, or had different kinds of insects, and because of these kinds of interactions, their beaks were exquisitely adapted. Darwin also looked at all kinds of other organisms and how diverse they were, and we know from work from a statement by Haldane, for example, that there's a huge diversity, for example, of beetles on this planet. Darwin had interesting ideas about what the mechanisms were, the cellular mechanisms by which these diversities arose. But he unfortunately got that aspect wrong. But the answer to how all this diversity arose came actually from another field. And that other field was really asking a completely different question. Darwin asked this question, how do the diversities of life on this planet arise? The other field was that of embryology, which asked this question, how are we made? And how are we made, as I said, is a question in embryology, but that came from linking another area, genetics, with embryology, and linking molecular biology, a much more recent field, with embryology. And not only did that answer this question on how are we made, but it answered the question, where do we come from, because it unraveled the shared chemistry. So what is genetics? And that field, again, I don't have time to go into detail, flowered, as it were, with the propagation of the work by Gregor Mendel. And Mendel, as you all know, studied variation from generation to generation in garden peas. But he was also studying at the same time all sorts of other characteristics in plants, height and weight and leaf size and so on and so forth, as were other people looking at how these quantitative traits were propagated from generation to generation. And they ended up in a mess. They couldn't find simple rules. But Mendel hit upon his simple rules by actually looking at inbred strains and mating them with each other and developing the rules which we now know today as Mendel's laws. Now, the challenge, of course, in genetics was that it told you what, from the outside, looking into a black box, what the rules were for simple characters going from generation to generation. Did it tell you anything at all about how the animal itself or the plant was made? And many critics of genetics at that time said, genetics can tell you how the rules by which the bristle on the back of a fly is positioned, but it cannot tell you how the fly itself is made. And that was a problem at that time with genetics. So let's go now linking genetics to the next aspect, which is embryology. And that's a question which has fascinated people for literally thousands of years. How are we made? And when people realize that we are made from an egg, a fertilized egg, which gives rise to a human embryo and which is amazingly born in a manner whose form is absolutely stunning, and that complex form comes from that simple embryo, people had all sorts of theories. One theory was that actually, and this is a very male-dominated patriarchal theory, that inside the sperm was a little human. And when that sperm mated with the egg provided the nutrition for the little human to become a big human. Of course, there's a slight problem, as if this was the case, then where did that little human in the sperm come from, right? And that came from another little human inside another sperm there, and so on and so forth, right? So this is a slight problem. The other view was that inside the egg, there were components which segregated asymmetrically as this fertilized egg divided into 2 cells, then 4 and 8 and so on and so forth. And somehow these components provided rules by which different parts of the animal, in this case, could arise. So when you had 1 cell and it divided to give rise to 2, you had some components going to this cell, other to this, and this made the left half and this made the right half. And then this divided again and it made 1 quarter and the other quarter and so on and so forth. What were the rules by which these components segregated? Who put these rules, these components inside, and how do they know that they have to go this way and that is a problem, because in some ways this is very similar to the problem of having a little human inside a sperm, right? So these fundamental issues, even though they had aspects of trying to answer mechanistically what was happening, ended up in a serious problem because we didn't have a mechanistic handle, a molecular handle of what's happening. People did all sorts of interesting experiments. They said, if that cell is dividing to give rise to 2 cells, and this cell knows I have to make the left half and this one the right half, if I kill this cell, then will this make only the right half? And indeed it did. When Willem Roos did that experiment, with a hot needle, he killed 1 cell, and he found that he got a half a tadpole over there. Hans Driesch did this experiment in another way. He took a sea urchin at the 2-cell stage and separated these 2 cells and kept them alive, and found amazingly a very dramatically different result. This half made an entire sea urchin, and this other half made another entire sea urchin, as if somehow this cell knew, quote unquote, that this other cell was no longer in contact. And this really shocked Hans Driesch, and he gave up biology and turned to religion and philosophy. So science can be dangerous. What actually was happening? The answer to these kinds of questions was that these cells are talking to each other came from experiments by Hilde Mangold and Hans Spearman. Hilde Mangold was a technician in Hans Spearman's lab and sadly died in a kitchen accident when she was very young, but Hans Spearman went on to get the Nobel Prize for this work. What Spearman and Mangold did was to take groups of cells from one region of a developing embryo and take these groups of cells and transplant them into another embryo in another region far away. Now, what can happen? These cells normally would have made one structure. They could still make the same structure when transplanted elsewhere, or they could respond somehow and figure out that they are in a new place and make structures corresponding to the new place, or perhaps they'll just die. But something dramatically different happened. These cells, when put in the new place, transformed that entire region near where they were into what would have been in this surrounding region. They had an influence on surrounding cells. And you can see that in this beautiful movie, and that's Hans Spearman on the left and Hilde Mangold on the right. And this experiment of Spearman and Mangold is being repeated by Eddie D. Robertus of the University of California at Los Angeles.